Okay, this is chapter 16 from my book, Medical Reformation, Vegan Renaissance Bible here. And we're gonna make this in two parts. So 16a, and then I'll do a separate lecture called that 16b. So what we're gonna talk about first of all is leaky gut, the mechanism of leaky gut, and we'll also go through the concept of dormant bacteria, bacterial endotoxin. And that'll be 16a. In chapter 16b, then I'll start going through the mechanism by which it causes blood clotting. It induces something called amyloid type clotting, amyloidogenic clotting. And that's relevant not only for leaky gut and for autoimmune disease type patients, but it's also relevant for viral infections, post-viral infections. The viruses can induce a prothrombotic, promote clotting in a way that can be harmful to health. And I think that's one of the explanations for some of the mysterious cognitive problems people have and brain fog. And, these small clots can lodge in the heart of the brain. I believe that's part of what's going on. But anyways, I'll talk about that in part B. Okay, for right now, let's go through leaky gut. This is part 16A. So here it is. Here's my drawing. This is a good one. You might want to hit your print screen button to save this one. So what it comes down to is, like I said, the internal medicine textbooks, medical school textbooks, rheumatology textbooks, they're all a joke. In my experience of looking at them, I don't even see leaky gut mentioned in the chapter. It's the most common cause of autoimmune disease. Okay, and the typical thing that's going to cause leaky gut is a lack of dietary fiber. So somebody's eating a meat and processed food primarily type diet. But there's a lot of other things that can do it. If you're taking antibiotics, those can wipe out the good bacteria. Oils are toxic to the good bacteria. Animal foods don't have any fiber, so they deplete the gut of fiber needed by the good bacteria. GP, glyphosate, glyphosate, it's sprayed on the GMO soy, and it's sprayed on other non-organic crops as well, including beans, including oats if it's not organic. That was originally a type of an antibiotic, and so that can be toxic to the good gut bacteria. Some other herbicides can be similarly toxic. Excessive psychological stress, I think it's because of vasoconstriction of the gut, the splanchnic circulation to the gut can cause it to have problems and have leaky gut. Caffeine will increase the risk of leaky gut. Alcohol increases leaky gut. Emulsifiers, those are chemicals to sort of stabilize processed food, to sort of hold the aqueous water phase in contact with the hydrophobic lipid phase, fat phase. Just being fat in general increases the risk of leaky gut. Having diabetes increases the risk. Polysorbate, these are some chemical additives to processed foods. Carrageenan. BT is Bacillus thuringiens corn, and that was the bacterial chemical related to causing insects' guts to pop. Well, it can irritate human guts too. That's in your GMO corn. High fructose corn syrup increases the risk of leaky gut. Some medicines do, like NSAIDs, non-steroidal anti-inflammatory drugs, like Moprin, ibuprofen, all those types of NSAID drugs. ASA is acetylsalicylic acid, just a fancy way to say aspirin. Increase with PPIs, antacids. Dishwasher soap can lead to these things. There's a lot of things. BPA, bisphenol A, the hard plastics. SIBO, small intestinal bacterial overgrowth. Gluten protein in some people. Oral contraceptive pills increasing candida risk as well. Artificial sweeteners can do it. Phosphoric acid in some of the soda pop drinks. Aluminum, mercury, radiation therapy, chemotherapy. Traumatic brain injury increases blood-brain barrier permeability. Things that increase blood-brain barrier permeability will increase gut permeability. Stroke will do it as well. And I am trying to rewrite the post-stroke, post-traumatic brain injury guidelines, and I've talked to a bunch of neurologists about this. They should be feeding patients higher fiber diets to help maintain that blood-brain barrier in the post-infarct, post-TBI injury phase, so that less toxins can get from the blood into the brain. Because you have a bad situation when a person has a stroke or they have a traumatic brain injury, they get increased gut permeability simultaneous with increased blood-brain barrier permeability, such that a toxin that gets through the gut layer can have increased access to the brain. Because you have a focus of destroyed brain in the center. Imagine this is your destroyed brain, and then in the periphery of that you have what is called a penumbra, the surrounding adjacent brain, and that means at risk. Penumbra is like Greek word for twilight. And so that brain could go either way. It's dysfunctional, but that could be temporary and it could restore to normal function, or it could go further down the path of destruction and die. So you want to protect that brain, and one of the ways you would do it is having increased fiber in the diet to maintain your tight junctions. Okay, so why does it work? Because these smiley face bacteria are the good bacteria. And there's really only two types of gut flora. You don't have to drive yourself crazy. They try to make it complicated. People are trying to sell you stuff. Okay, so basically there's good gut bacteria that comes from eating fiber and living a sort of a natural traditional lifestyle like Adam and Eve, but keep your indoor heating and plumbing. And then there's bad gut bacteria, and that comes from eating processed food, no dietary fiber, low dietary fiber, and all these other things in here. So the good gut bacteria with the smiley faces, they eat the dietary fiber. They convert it into short-chain fatty acids, SCFA. There's 2-carbon acetate, 3-carbon propionate, and those just go through the portal vein. They go up to your liver, okay? And the liver uses them to make fatty acids, and the 2-carbon ones to make even number of fatty acids, which is most fatty acids. And then the 3-carbon enables them to make odd chain on the rare occasion when they want to make those. 4-carbon fatty acids, the butyrate, those go to the enterocytes. The gut's the enteric tract, so these are the enterocytes, the lining cells of the gut. It's only a single cell thick, and it uses them to make these TJs. TJ here stands for tight junctions. So the tight junction means that nothing can get in between these cells. There's a few other cells in here, like immune system cells are all in green here, all right? And then there's the blood vessels right beneath there. Okay, so here's the enterocyte layer, single cell layer. Here's the tight junctions. There's a mucus layer, an inner and an outer mucus layer. They have secrete some antimicrobial peptides in there, some immune globulins in there. All right, all of that is protecting your gut barrier. And the point I'm making is these things all damage your bacteria. They either damage the lining or they will damage the bacteria. So the bottom line is when you don't have fiber and you can't make your butyrate, you don't make your tight junctions, and now things that should not get through the gut are getting through the gut. These are the bad bacteria here, and they've got a big frown on their face. These types of bacteria can eat the mucus layer, and that can lead to then subsequent damage of the intestinal lining, the enterocytes. Also, they make an endotoxin. The gram-negative bacteria make LPS, lipopolysaccharide. Gram-positive bacteria make something called LTA, lipoteichoic acid, and they can get through the gut lining because the tight junctions are not intact, and they can incite an immune response in the lamina propria immediately deep to these cells here. And they can also get into the blood, the LPS, for example, and it makes the blood more prone to clotting. So it increases blood clotting. That's a bad thing. You don't want it because we talked about this before. In the movies, people die from bleeding. In real life, they mostly die from clotting, okay? Almost everybody is. Clot in the brain, it's a stroke, okay? A clot in the heart, it's a heart attack. A clot in an artery that supplies an important organ, it becomes ischemic, and it can then subsequently develop cancer. So it's a major contributor. Lots of morbidity in older people is all due to clotting off arteries, okay? They clot off the arteries in their eyes, they can't see. They clot off the artery to the nerve for hearing, they can't hear. Okay, they clot off other arteries in their body, and they're just weak. Okay, anyways, the other thing is normally you can only absorb about a tripeptide or less, meaning 3 amino acids connected together. But when you have a lack of tight junctions, you can now absorb bigger chunks of protein, more than 3 amino acids. You can get a whole big piece of protein. And when it comes from an animal, it can have an amino acid sequence very similar to that in the human. And what will happen is it'll be different enough that the immune system will react to it. So here's an immune system formed antibody. It looks like the letter Y, and that antibody binds to it. But then it's different enough that that same antibody might cross-react with our own body once it's made in large amounts, and that's autoimmune disease. That's the most common mechanism of autoimmune disease. So it's called molecular mimicry in that the animal protein ingested that got across the gut lining mimics those of our own. But it still forms antibodies because our body is able to recognize it as foreign. Then there's cross-reactivity with our own body because it's similar enough that that same antibody will react with our own bodily tissue. So that's called, leaky gut or increased intestinal permeability with subsequent molecular mimicry, autoantibody cross-reactivity. You sometimes hear the word PAMPs and DAMPs. Okay, PAMPs means pathogen-associated molecular patterns. Let's see here. What else do I want to talk about? I covered most of this when I was going through all that. Okay, here we're going to talk about the whole process. I got all the words here for you if you want them. Leaky gut, absorption of abnormally large pieces of protein. Immune system recognizes a protein chunk as foreign. Antibodies are formed to it. The animal protein is similar enough, molecular mimicry. Confuses the antibody. Auto means to attack itself. Cross-reactivity, okay, fine. Dairy is thought to be the main cause, let's say, of diabetes, and it can damage pancreatic beta cells. Dairy is also thought to be probably the main cause of multiple sclerosis, like butyrofalin, autoantibodies cross-reacting to that. There was more MS in the high dairy areas, let's say in Norway, than Roy Swang pointed that out many, many years ago. Okay, this new stuff, Hashimoto's, is bad too. Some people think too, like McDougall will give you a lecture and he'll say you eat a hot dog, which is a ground up animal, and there'll be some thyroid ground up in there, and then you can get the same problem in the context of leaky gut with autoantibodies to the thyroid tissue cross-reacting with your own tissue, and he thinks that's the main reason to cause Hashimoto's thyroiditis. Glyphosate, I'll just abbreviate it GP from here on out, can damage, can lead to gut intestinal lining, leaky gut. It can also have a damaging effect on sperm, according to Stephanie Seneff, PhD from MIT. She's the one who wrote the book Toxic Legacy. I even referenced some of the page numbers to that book here in this talk. Okay, now I'm going to talk about something related to GP, and this is a little bit interesting. It's relevant. So here's your basic amino acid. Your basic amino acids has a alpha. This is the alpha symbol. Alpha carbon in the center of it. It's called amino because there's an amino group on one side of the alpha carbon. And it's called amino acid because there's an acid, a carboxylic acid here, which means a carbon group with a double bond to oxygen and also a single bond to a hydroxyl group or an alcohol group. And so that's called a carboxylic acid. So amino acid. Okay, the alpha carbon then always has a hydrogen sticking up, and it's like a head, you know. And I said, too, this reminds me of Jesus Christ on the cross. Okay, there's the head right there. And there's one arm would be an amino, and another arm would be the carboxylic acid, and then the legs going down, that's the R group, that's the variable. And you can often just write it, alpha R, because R is the only thing variable in there. Okay, let's see, we just covered all that. Now I'm going to get in a little more detail about the difference between glycine and glyphosate, or glyphosate, or glycine phosphate. A lot of times some people have corrected me and said, oh, you should be calling it, you know, glyphosate, that's what its real name is. You shouldn't call it glyphosate. Well, when I do something, a lot of times it's for a reason. I'm trying to help the audience to learn it better. So here's a glycine. There's the alpha carbon. And what's unique about glycine, it's the smallest amino acid. Its R group is just a hydrogen. That's as small as it can possibly be. So it's the smallest possible amino acid. Glyphosate is unique in the sense that instead of just having an amino, it has an extra phosphate stuck on here. So just think of it as glycine phosphate. There's a little more to it. There's an intervening carbon, a methyl group, but just think of it as a phosphate. That'll get you what you need for understanding what's about to be shown to you about how it has a negative effect on enzymes. So glycine is the smallest amino acid, and because it's so small with just a hydrogen for its R group, it'll often be found in an amino acid, in an enzyme active site, because it creates space in the pocket by being so tiny, allowing the substrates to come in and fit better into the pocket. It's also useful on the inner surface of an ion channel to make more room for stuff to enter a cell. Okay. And it can substitute into proteins, according to Stephanie Seneff. She wrote an entire book about it, again, called Toxic Legacy. And she thinks that explains a lot of diseases associated with GP. It's substituting for glycines and proteins and then causing the proteins to be damaged. They could be misfolded, and when they're misfolded, they can't carry out their normal functions. And it can lead also to dysfunction in the immune system. She thinks it's distorting the complement protein C1q. I even referenced it right there in her book. Okay, so a little bit about what does an enzyme do. An enzyme lowers the activation energy for a reaction. So let's say normally it's hard to run a reaction because you need tons of energy invested in the system in order to get it to be able to go to product. What an enzyme can do at its active site is lower the transition site energy such that this reaction be much more likely to progress to its completion with the formation of the products. And the enzyme functional process has been likened in the past to a lock going into a key. And then they realized, well, the enzyme changes shape a little bit when it binds to the substrate. So then the next model of describing that was, well, it's like a hand going into a glove. That's a reasonable idea. So what the enzyme does is it puts the two substrates and it gets them to be close together and orientated such that they can react with each other. So I showed the green thing going into the red thing here. All right, and then they break apart and you have new products. So these are the substrates, the preliminary materials. There's the energy transition state in the enzyme, and then there's your products coming out over here. The hand in the glove is called the induced fit model. Okay, and then here's something kind of cool, that you've got also other things the enzyme can do to get the substrates to come into the enzyme active site. So it can be like a vacuum. So imagine you had a negatively charged, or a magnet, okay, you had a negatively charged substrate. And then to pull it towards the active site, you can have positive charges in here. So let's say these are all weakly positive, and that substrate's just sucked in here like a vacuum or like a magnet towards the active site that's even more strongly positively charged. So I thought that was kind of cool. You see this kind of stuff all the time in medicine, molecular biology, and biochemistry, that there's like God designing the body is just a genius. It's amazing. You know, people tell me, oh, why do you believe in God? Is that sure? I'm like, because I study this stuff, and it's just so far beyond what a human brain is capable, you know? It's like basically if you were walking at the beach and you saw your name written down, you would know an intelligent person who was literate had done that. So how does 3.3 billion DNA base pairs write itself? It doesn't. That knowledge and information has to come from somewhere, and it's not going to spontaneously happen. Okay, so first of all, here's an enzyme where the active site is perhaps a little too big. And you'll have the substrates, but they're not orientated properly to react. Here's a second enzyme, and its active site is just right. The substrates are properly orientated so that they can react with one another, and the action will enzyme, and it'll proceed to completion. Okay, very common. If you have a negative charge inside the active site, it'll attract a positive charge substrate to come into it. Okay, now here's showing that, let's say a regular enzyme active site has a amino acid alanine that just has an R group of like a methyl group, and here's a glycine, it's R group, it's just a hydrogen. So there's a lot of space for a substrate to come in here. Now if you substitute glyphosate, GP, for glycine, it's going to be a lot bulkier because it's got that big phosphate group, and this phosphate group has a negative charge. So if let's say the substrate was a negatively charged something, it's going to be repelled by that negatively charged phosphate. But the big thing here is just the bulkiness of the phosphate is going to take up a lot of space in the enzyme active site, make it hard for the normal substrate to bind to it. Glyphosate can substitute for glycine, and it can damage collagen, because in collagen, every third residue is a glycine. So if you get this glyphosate substituting for that in multiple spots, it's going to distort the shape of it, because you have these individual fibers of collagen, and then they wrap around each other, three of them, into a triple helix. So if you got the initial baseline fibers all distorted by glyphosate for glycine substitutions, the whole thing's not going to hold together very well. And this is what the research that Stephanie sent us says is she's pretty sure is going on. Like I said, every third amino acid in collagen is glycine. And collagen is super important. It's your connective tissue. It's about 1/3 of the proteins in the whole human body. It's the most common protein in the human body. So if that's getting messed up with GP, you got a real problem on your hands. Okay, lots of people go for hip joint replacement, knee joint replacement, and part of it, can be due to dysfunction of the collagen. And there's a couple things that tend to disrupt collagen synthesis. It could be due to ischemia. That's a ubiquitous problem in the human body in Westerners, middle-aged and older. It could be due to F minus disrupting collagen synthesis in the water, fluoride. That'll predispose to problems. Okay, it can be GP, glyphosate, disrupting collagen synthesis. Scurvy, a lack of vitamin C, can also contribute to collagen synthesis problems. Other things too, AGEs, advanced glycation end products associated with diet or diabetes, can make things a little worse in some rare disorders. So anyways, these are all things that can make your collagen disrupted, and they can lead to spinal degeneration. All these things can contribute to that. All right, this next part is really part of the second topic, so I think we're going to end there. So this will be the end of chapter 16a. We'll start up with this part because it's going to get into that amyloidogenic clotting, like Douglas Cowan, atherese, pretorus work. That'll be 16b. We'll do that at a later time. So anyways, I hope this stuff was helpful, and the main stuff we covered for today was just leaky gut, the causes of it, and also the way GP can substitute for glycine and damage proteins, including super important proteins like collagen. So, hope that was helpful.